Hello students, in this video lecture we are going to learn about the third chapter of cell biology, that is chloroplast, mitochondria and the endoplasmic reticulum. So in this video lecture we will learn about the three different cell organelles of plant eukaryotic cells, that is chloroplast, then mitochondria and the endoplasmic reticulum, right? So first we'll start with the chloroplast. So in this photograph you can see these are the leaves, the green leaves, right? And if you take the transverse section of these leaves, then you will get the structure, whatever you can see here, the leaf in the leaf anatomy, right? Anatomy means the internal structure of leaf. And in this leaf anatomy you can observe different kinds of cells, right? Tissues as well as different kinds of cells. So we will select one green cells here, right? So these green cells, as they perform the function of photosynthesis, right, it means they are having the chloroplast. So this chloroplast you can see here. So this chloroplast having the different components like the granules, right, stroma, then thylakoids, etc. So and this chloroplast is involved in the function of photosynthesis, that is called the synthesis of food material from the carbon dioxide and the water by utilization of the light energy, right? And just it will convert into the chemical energy in the form of carbohydrate, that is glucose, right? So let's start with the chloroplast. The term chloroplast was first described by the scientists Nemia Grave and the Antony van Leeuwenhoek, right? So the chloro means the green, while the plast means living. So something which is green in color and it is living things. So that is the chloroplast. So this chloroplast having the chlorophyll pigments present inside this impact green colored which is present inside the green cells of plants. So these chloroplasts are present in the plants, other eukaryotic organisms that conduct the photosynthesis, right? So here you can see the structure of this chloroplast. So these chloroplasts, they are plano-convex lens-like structure, which is about 5 micrometer in diameter and 2 to 3 micrometer in thickness, right? So this chloroplast, they comprises of the or they get protected by the two different layers, right? It means it's a double layered envelope cell organelle. So these two different layers are called as a outer membrane and inner membrane. So outer membrane is at the outer side and inner will be at the inner side, right? Then the granular stroma, you can see here the granum, right? So which consists of the stack of thylakoid, as like the tiffin, right? So we have the tiffin and there are different boxes. So each plate act as a grana. So this grana get arranged one to each other by vertically, and thus they form the structure of granum. So granum is single grana. In this chloroplast you can observe here there are many granum, okay? So these are called as a grana. So if there are two or more than two in number, we will call it as a grana, and if it will be single in number, then we will call it as a granum, right? So here you can see the space in between the outer membrane and inner membrane. So such kind of space is called as a intermembrane space. Intermembrane means the space in between the two different membrane, okay? Then you can see these thylakoids, they are arranged vertically, they form the structure of grana. There are many grana arranged inside this chloroplast, okay? And you can see here the gel like substance as like the cytoplasm in the cell, so that is called as a stroma, right? So this chloroplast, as they are involved in the synthesis of food material, means they perform the photosynthesis, so thus they are having the chlorophyll pigment. So these chlorophyll pigments, they are present inside the single thylakoid, right? So you can see the plate-like structure. So this plate-like structure is called the thylakoid, and this thylakoid contains many chlorophyll pigments. So these chlorophyll pigments are of different types in the plant kingdom, like the chlorophyll pigment A, chlorophyll pigment B, then C, D, then chlorophyll pigment E, xanthophyll, pheophytin, as like that, right? So there are different kinds of chlorophyll pigments. So it depends on the species of the plant. If in the different plant species there are different kinds of chlorophyll pigments. In the algae, right, some algae are red in color, some are brown in color, so there are different kinds of algae. So this coloration will be due to the presence of different kinds of chlorophyll pigments, which is present inside this thylakoid membrane, right, inside the thylakoid. So these chlorophyll pigments, they absorb the light, right, which is bombarded on the leaf. So this light get entrapped by this chlorophyll pigment, and thus it get utilized for the generation of ATP, and this ATP get utilized during the photosynthetic reaction, which will be takes place in the stroma, right? So remember that the light reaction means the it's a kind of the photosynthetic reaction. So photosynthesis, it's divided into two types of reaction: light reaction and dark reaction. So light reactions, they are dependent, and dark reactions are called, they are light independent. If there will be light or no light, then also this reaction will be takes place. So that's it, these are called as a dark reaction. So light reaction will be takes place inside the grana, and dark reaction takes place in the stroma, means the food will be get synthesized in the stroma, right? As you know that the chloroplast, it's one of the semi-autonomous cell organelles, so that's why it's having its own genetic material, it's having its own DNA, so that is called as a chloroplast DNA, right? And this chloroplast DNA is circular. If you observe the DNA of any higher organism, so they are having the helical structure, right, thread-like structure, but this DNA, that is the chloroplast DNA, is circular in shape, right, as like the bacteria. So here we can see the envelope of this chloroplast. So this envelope, it's composed of the system of double membrane, each of which is near about 50 to 80 angstrom unit thick, and has a unit membrane structure. The outer membrane is separated from the inner membrane by the intermembrane space, and its diameter is near about 10 nanometer, right? So here you can see the outer as well as inner membrane and the intermembrane space in between this chloroplast. Then next component of the chloroplast is the stroma. So here you can see in this picture the gel-like semi-solid substance that is called the stroma, right? So this stroma fills most of the volume of the chloroplast, as it contains about 50% of the proteins of the chloroplast, right? So you know this chloroplast having its own genome, its own DNA, so that DNA can be get transcribed as well as translate to form the proteins, and these proteins are just floating on the stroma, right? So it contains the ribosomes as well as DNA molecules also, as is having its own machinery. So that's why it's called as a semi-autonomous cell organ. Semi means half. So it can synthesize its own proteins, but not 100%. This chloroplast is depends on the nucleus also for the requirements of proteins. So that's why it's called the semi-autonomous cell organelle. Then the thylakoids. So these thylakoids, it consists of the flattened and the closed vesicles, which are arranged as a membranous network. So in this photograph you can see, right, the plate-like structure. So it may be stacked like a net pile of the coins, which forming the grana, right? So there are maybe 40 to 80 grana in the matrix of the chloroplast. And the thylakoids, they include the light absorbing pigment, as I just told you earlier, right? So these light absorbing pigments, they are present inside the thylakoid, and a complex chain of electron carriers and an ATP synthesizing apparatus is uh, present on the thylakoid membrane, right? So this is about the thylakoid. Then we'll see the chloroplast DNA. So this chloroplast DNA, that is also called as the cpDNA, right? So it's also known as the plastid DNA also, that is ptDNA. Both the meaning are same. This chloroplast DNA is circular double stranded DNA molecules, right? So as like the bacteria, they are having circular DNA which is double stranded, and this chloroplast genome size is near about 120 to 270 kilobytes, right? The majority of the plants, they fall into 120 to 160 kilobytes, but some of the plants, their chloroplast genome size is near about 270, right? So this is about the chloroplast DNA. So they contains about 100 genes to synthesize their proteins, right? So their DNA, the chloroplast DNA, having 100 genes to synthesize their proteins. And this chloroplast DNA regions, they include the large single copy and small single copy regions, and they show the inverted repeats, means the nucleotide sequences, they just get repeated. Okay, so we will learn about this, what are the nucleotides and what the DNA sequences in the molecular biology of this paper, right? The conifers and the group of the legumes, they lack the inverted repeats. Conifer means these are the members of gymnosperm, and the legume plant means what? They develop the pods, right? We will call these legumes in Marathi like the chenga, right? So they doesn't have such kind of the inverted repeats. And then what are the functions of chloroplast? So it's very clear that these chloroplasts, they perform the functions of photosynthesis, right? So this chloroplast is the site of photosynthesis. The light reaction as well as the dark reaction of this chloroplast takes place inside the light as well as dark reaction of the photosynthesis takes place inside the chloroplast. So light reaction will be takes place in the thylakoid membrane, and the dark reaction will be takes place in the stroma, right? So here you can see the Kelvin cycle. It takes place inside this chloroplast, and there are some enzymes, right? That is called as the Rubisco enzyme, ribulose bisphosphate carboxylase enzyme. It's present in the chloroplast, so it fixes that atmospheric carbon dioxide and convert it into the sugar, right? That is glucose. So this reaction, that is the dark reaction, takes place in the stroma, and the light reaction means what? The light dependent reaction. And here what will happen? Whatever the water which is present inside the chloroplast, it will be get breakdown, and thus it forms the H plus ion. So this is the main motto of the light reaction, to generation of the H plus ion, to generate the protons. So this proton is get utilized for the generation of ATP, and again this ATP get generated in the light reaction, so the ATP will be get utilized in the dark reaction for the synthesis of food material, right? So this is the major function of this chloroplast. Then we will see the next cell organelles. You can see in this photograph, this is the mitochondria, right? You can guess easily, as you can observe its shape. The inner membrane, it just shows the inner folding, right? So on the basis of that, you can easily you can guess that this is the mitochondria. So this mitochondria is also one of the double membrane cell organelle, as like the chloroplast as well as nucleus. You know, its famous common name is the this powerhouse of cells, right? So in this photograph, you can see the different components of different parts of this mitochondria, right? So we will discuss it in the next slides, right, in detail. So first we'll introduce this mitochondria. So this is mitochondria. It's a Greek word. It is a double membrane bound organelle which found in the eukaryotic cells. Prokaryotic cell doesn't have mitochondria, right? So mito means a thread and the chondrion means a granule. So granule like threads which are present in the eukaryotic cell, so that is called the mitochondria. So these are found in the cytoplasm of the cells. As you know that all the cell organelles, they are in the eukaryotic cell, they just float on the cytoplasm, right? So as it's commonly known as a powerhouse of cell, and you know why it's called powerhouse of cell, because it generates an energy rich molecules, that is ATP molecules, adenosine triphosphate. So the first discovery of this mitochondria, or first this mitochondria is observed by the scientist Richard Altman in 1894, and this mitochondria term was first coined by the scientist Carl Benda in 1898. Okay, just observation of this mitochondria after the 4 years, right? So the theory, the endosymbiotic theory says that there is a inner folding in the plasma membrane of an ancestral prokaryotic cells. They give rise to the endomembrane components, which including the nucleus as well as the endoplasmic reticulum, right? And these proto-eukaryotes, that is prokaryotic cells, but it just getting evolved to becomes the eukaryotic cells, right? So that's why this cell will be called as a proto-eukaryotes. So in the first endosymbiotic event, the ancestral eukaryotes consumed aerobic bacteria that evolve into the mitochondria. So in this way the mitochondria get evolved, right? In second symbiotic event, the early eukaryote, they consume the photosynthetic bacteria that evolve into the chloroplast, right? And in this way the eukaryotic cell get evolved which having the mitochondria as well as the chloroplast, and there will be a symbiotic association in between these two different kinds of bacteria. I will call it mitochondria as well as chloroplast as a bacteria, because why? Because they shows the characteristic of bacteria. They have the circular DNA, they have the double membrane structure, and if you observe the nature of this membrane of mitochondria as well as chloroplast, then they are exactly same as like the plasma membrane, right? So that's why we can easily hypothesize that these are the once upon a time there was the free living bacteria, right? So in this way the modern heterotrophic eukaryotic cell get evolved as well as modern photosynthetic eukaryotic cell get evolved, right? Heterotrophic eukaryotes means they have only mitochondria but not the chloroplast, so that's why they cannot synthesize their own food material. But the modern photosynthetic eukaryotes, they have the chloroplast as well as the mitochondria, right? So we will call it as a plant cells, and modern heterotrophic eukaryotes we will call as a animal cell, because animal cannot synthesize their food but plants can. So that's why we will call the photosynthetic eukaryote of plant cells. So in this way the endosymbiotic theory, this says that the eukaryotic cells they also get evolved from the prokaryotic cell, right? As you know the prokaryotic cell is more what we can say it's like the ancestor of the eukaryotic cell, right? The morphology of mitochondria says that the mitochondrial size it ranges from 0.5 to 1 micrometer in diameter, right? It means if you compare the size of mitochondria and the chloroplast, then you will get the idea about the size of both. Mitochondria is smaller than the chloroplast. The shape is normally sausage shape, right? It's elongated as well as oval type. So in the fibroblast cells, it's get elongated and looking like a thread. The number of this mitochondria is depend on the type, size, and the functional state of the cells. For example, an average liver cells they contains around 1500 mitochondria, right? Because the liver it's very important glands which is present in the animals. They perform the different kinds of functions, right? So that's why they need more energy, and thus the number of the mitochondria in the liver cells is also higher. Where is the located this mitochondria, right? So the cells with the high energy requirements they will get, right? For example, the sperm tail, the muscles, as well as the flagella, they are having the mitochondria because of the mobility, because of the continuous metabolic activities. Then we'll discuss about the structure of these mitochondria. The mitochondria having five different kinds of components, like the as like the chloroplast, they are having outer membrane, inner membrane, then intermembrane space in between the outer and inner membrane, then cristae, then matrix also, right? So we will see it in detail. So in this picture, in this slide, you can observe the structure of this mitochondria, as it shows the outer membrane which is present at the outer side, inner membrane which is present at the inner side, right? And there is a space in between these two membrane, right? So that is called as a intermembrane space. So this thing is common in both chloroplast as well as mitochondria, right? But there is some speciality in the mitochondria. The inner membrane shows the inner folding, so that's why it looks like zigzag manner, right? So this is the inner membrane, and inside this inner membrane the gel like substance is there as like the cytoplasm of the cells, so that is called as a matrix. Okay, and is one of the this mitochondria was once upon a time free living bacteria, so it's having its own genetic material that is called the mitochondrial DNA, or it is also called as a mtDNA, right? And as they have their own genetic material, that is the DNA, so they also having the ribosomes, so that they can synthesize their own proteins. But again, as like the chloroplast, the mitochondria is also one of the semi-autonomous cell organelle. Means these mitochondria, whatever the requirements of the mitochondria for the proteins, so all the proteins, it cannot be synthesized by its own DNA. It depends on the nucleus of the cells also. So that's why it is called semi-autonomous cell organelle. Semi means half, right? So in this picture we have also observed the F1 F0 complexes, right? So these are the components of ATP synthesis. So where the ATP will be get synthesized. The outer membrane, it's a simple phospholipid bilayer as like the cell membrane, right? So it encloses the mitochondria, the content of the mitochondria. It containing the protein structure that is called the porins, right? So these porins are the proteins which is present on the outer membrane. Then the ions, nutrient molecules, then ATP, ADP, etc. So these ions, nutrient molecules, ATP, ADP, they can be passed through this outer membrane with ease, right? That doesn't need of any kind of special energy to transport this, as they are having the porins, right? So this porin act as a transporter. Then the inner membrane. So inner membrane is freely permeable only for the oxygen, carbon dioxide, and the water, right? So they contain the complexes of the electron transport system. You heard about the electron transport system. In the electron transport system, water molecule get break down to generate the photons, and these photons get utilized to generate the ATPs, right? Then ATP synthase complex. So whatever in the electron transport system, the photons will be get released. So ATP synthase will utilize those photons to regenerate ATP from the ADP. So after the utilization of ATP, what will happen? ADP will be get generated, ADP and inorganic phosphate, and again this ADP and inorganic phosphate will recycle to convert it into the ATP. Okay, then the transporter proteins. The presence of these sophisticated ion transports, they exist on this inner membrane of the mitochondria. The several antipode system, they also exist on the inner membrane, which allowing the exchange of anions, right, which between the cytosol and the mitochondrial matrix. Okay, so it is devoid of the cholesterol and rich in the phospholipids and cardiolipins. So this is about the inner membrane. So here you can see the inner membrane space, the outer membrane, inner membrane, and at the inner mitochondrial membrane you will find the electron transport system, ATP synthase, right, and inside that matrix. The intermembrane space, it's a cavity between the inner membrane and outer membrane, right? It has high proton concentration. So the concentration of the protons is highest in this space, that is called the intermembrane space. The space between the intermembrane and the outer membrane is size is approximately 70 angstrom unit. The concentration is same as that of the cytosol. Okay, then you will see the next component, that is called as a cristae. So these are the folds of inner mitochondrial membranes. They show the stacked like particles or inner membranes spheres. The cristae is covered with this inner membrane spheres, that is called the stalk particles or knobs, or it also called as a heads. So they contain the proteins called F1 portion and F0 portion for the ATP synthesis and the ATO oxidation. Then the next component, that is mitochondrial matrix. So this mitochondrial matrix is a gel-like consistency inside this mitochondria. This matrix, the mitochondrial matrix, is dense and homogeneous in nature, and the 2/3 of the total proteins of the mitochondria is just floating on this matrix. So mitochondrial matrix, they have the enzymes, ribosomes, DNA, mRNA, granules, fibrils, tubules, all these are floating on the matrix. The major enzymes, they include the enzymes involved in the synthesis of nucleic acid, that is this DNA replication, as well as transcription process where the RNA gets synthesized, and the protein synthesis, that is translation. The fatty acid oxidation enzymes, they are also exist inside this matrix. And the TCA cycle. So TCA cycle, it's a Krebs cycle, right? And this cycle, in this cycle you can say that the generation of the ATP and ATPase2 molecules get generated here in the TCA cycle, right? And these are the biochemical reactions. So that's why the biochemical reaction need an enzyme, and these enzymes are also exist in the mitochondrial matrix, except the succinate dehydrogenase, and remaining all the enzymes, they are present inside this matrix and synthesis inside the matrix also. Okay, then we'll see about the mitochondrial DNA, that is mtDNA. So this mtDNA or the mitochondrial DNA, it is a double stranded covalently closed circular molecules as like the bacteria, so which occurs in the multiple copies. Okay, mostly the remains attached to the inner mitochondrial membrane, that is called the cristae. They store the biological information which is required for the growth and the multiplication of this mitochondria. They can undergo the replication process, duplication process, okay, then transcription process, translation process to synthesize the proteins. The human mitochondrial DNA is also like circular in nature, not absolutely autonomous. It depends on the nuclear DNA also. That's why it's called partially autonomous or semi-autonomous cell organelle. Then we'll see the functions of mitochondria, and the common function, that is the synthesis of ATP. The energy introducer of the cells, that is the synthesis of ATP, will be takes place inside the mitochondria in the reaction of Krebs cycle in the matrix, as well as the electron transport system, as well as the phosphorylating the system, that is ATP enzyme. So here the ATP will be get generated. So mitochondria is also act as a extra mitochondrial inheritance. So chloroplast also involved in the inheritance of the characteristic. As chloroplast is also having its own DNA, mitochondria is also having its own DNA. Right? So this mitochondrial DNA contains the plasma genes, that is the extra chromosomal gene. They transmitted from one generation to the next generation, from the mother to the offspring. Right? So this synthesis of mitochondrial DNA, RNA, proteins, they brings about the gene expression through the gene duplication, transcription and translation process. They synthesize 13 different kinds of polypeptides in the human. Right? So this is the functions of mitochondria. Then again we will see another functions like the site of several metabolic reactions provided by the mitochondria. The outer membrane, in the outer membrane, the oxidation of epinephrine, degradation of tryptophan, elongation of fatty acids, that takes place inside on the outer membrane, while on the inner membrane of the mitochondria, oxidative phosphorylation will be takes place. Okay, and in the matrix, Krebs cycle, beta oxidation of the fatty acids, then detoxification of ammonia in the urea cycle, storage of the calcium ions is also the function of the matrix. Okay, other functions like the production of heat, like the non-shivering thermogenesis. As you know that the hot-blooded organism, right? So we can control our own body temperature by different kinds of mechanism like shivering and non-shivering thermogenesis. Then this mitochondria is also involved in the apoptosis process, means the programmed cell death. Then it's also involved in the role of the neurotransmitter metabolism as well as the cholesterol metabolism also. Right? So this is all about the mitochondria. Then we will talk about the next cell organelle, that is called the endoplasmic reticulum. So in this slide, in this photograph, you can see the structure of this endoplasmic reticulum. Right? So this endoplasmic reticulum is sometimes get integrated with the outer membrane of nucleus. Right? It is get integrated with the outer membrane of nucleus. Then we'll see this endoplasmic, it means within the plasm, and reticulum means the network. The endoplasmic reticulum is a network of what? It's a network of flat and vascular structures which like extends throughout the cytoplasm in the plants and the animal cell. Right? So these are the sacs and the tubules which are all interconnected by single continuous membrane, so that organelle has only one large, highly convoluted and complexly arranged lumen. So this lumen is called the internal space in this endoplasmic reticulum. So it takes up approximately 12% of the total volume of cell. If I am talking about the area occupied by the cell organelle, then the largest cell organelle in the eukaryotic cell is a endoplasmic reticulum. But if I am talking about the size, then always it will be nucleus. But the area of the cytoplasm of the cell get occupied more by the endoplasmic reticulum. So it is connected to the double layered nuclear envelope, right, the outer membrane of nucleus. So how is the endoplasmic reticulum get formed? What is the origin of endoplasmic reticulum? Okay, so at the present manner of origin of the endoplasmic reticulum is not definedly known, not clear cutly known, but the concrete hypothesis is that the endoplasmic reticulum is budded off from the nuclear envelope. So this has been postulated by the scientist F.Y.B.Sc, right, in the 1974. This endoplasmic reticulum appears to arise from the outer membrane of nuclear envelope by outfolding, right, or from the plasma membrane by infolding. Either it get formed from the outer membrane of nucleus or the plasma membrane of the cells. So either plasma membrane shows infolding to form the endoplasmic reticulum, or outer membrane of the nuclear membrane shows the outer folding to form the endoplasmic reticulum. The smooth endoplasmic reticulum seems to arise from the rough endoplasmic reticulum by detachment of ribosomes. Then we will see about the structure of this endoplasmic reticulum. The endoplasmic reticulum is three-dimensional network of intracellular. It is formed from three different kinds of elements, that is cisternae, tubules and the vesicles. Cisternae. So what is this cisternae? This is the flat, unbranched and sac-like element, right? They lie in stacks parallel to one another, and they bears the ribosomes on the surface, that therefore appears rough, right? So that's why such endoplasmic reticulum is having the ribosomes on their membrane. They are called as a rough endoplasmic reticulum. So it contains the glycoproteins like ribofurin 1 and ribofurin 2, so that binds the ribosomes. So here you can see in this photograph this endoplasmic reticulum. It shows the two different kinds of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum, on the basis of presence or absence of ribosomes. So as these ribosomes, they are small circular molecules, they attach to the endoplasmic reticulum. So that's why this endoplasmic reticulum looking like rough, right? So that's why such endoplasmic reticulum is called rough endoplasmic reticulum or granular endoplasmic reticulum. And if ribosomes are not attached to the endoplasmic reticulum surface of endoplasmic reticulum, then such endoplasmic reticulum is called smooth endoplasmic reticulum or a granular endoplasmic reticulum. These component of the endoplasmic reticulums are cisternae. In this photograph you can see the elongated structure. Then vesicles. These vesicles are either oval in shape or circular in shape. And the tubules, right? So these tubules are branched. Okay. So these tubules, these are the irregular branching elements which forms the network along with other elements. So these are often free of the ribosomes, right? means ribosomes are not attached to the tubules. Then vesicle. These vesicles are oval and round shaped bodies looking like vacuole of this cell. So these are also free of ribosomes. All the elements of endoplasmic reticulum freely communicates with one another, and they contain the fluid that is called the endoplasmic matrix, as like the matrix of mitochondria in the endoplasmic reticulum lumen. So this matrix is different from the cytoplasmic matrix outside the ER, outside the endoplasmic reticulum. So the ER, endoplasmic reticulum, they may pass from one cell to another through the plasmodesmata in the form of desmotubules. Okay, so in this way one cell can interact with the another cell through the plasmodesmata. In the previous lecture we discussed about the plasmodesmata, right? So through this plasmodesmata desmotubules can transfer. So this is the picture of endoplasmic reticulum which I have seen earlier. Then we will see the endoplasmic matrix. The space inside the tubules and the vesicle is filled with the watery medium, so that is different from the fluid in this cytoplasm or cytosol outside the endoplasmic reticulum. So their walls are constructed of lipid bilayers, the membranes that contains the large amount of proteins which are similar to the cell membrane. So first we will see the smooth endoplasmic reticulum. So this smooth endoplasmic reticulum is an arrangement of tubules, vesicles and sacs. The size and the structure of this SER varies between the cells. SER can change within the cell's lifetime to allow the cells to adapt to the changes in its functions and the requirement, right? So these are not having the ribosomes, right, to their membranes. So this SER is connected to the nuclear envelope. So the network of SER, it allows there to be enough surface area of the action or the storage of key enzymes or the products of the enzymes. SER is less stable as compared to rough endoplasmic reticulum, RER. The SER is characterized of the cells in which the synthesis of non-protein substances takes place. Then we will see RER, that is rough endoplasmic reticulum. So the surface of RER is studded with the ribosomes, which giving the rough appearance. So it mainly consists of cisternae, right? So cisternae are what? The elongated structure, unbranched structure. The membrane of this RER forms the large double membrane sheets, which is located near and continues with the outer layer of the nuclear envelope. So this rough endoplasmic reticulum is very important in the synthesis and the packaging of the proteins. For example, Russell bodies of the plasma, nasal granules of the nerve cells. So all these are get formed from the RER. The binding site of these ribosomes on the RER is the translocons. These ribosomes bound to the RER at any one time are not stable part of this organelle structure, because the ribosomes are consistently being bound and released from the membrane, okay, as they play important role in the synthesis of proteins in the cytoplasm. So ribosomes only bind with the rough endoplasmic reticulum once a specific protein nucleic acid complex they forms in the cytoplasm. Then and then only they will bind with the RER. So this special complex forms when the free ribosomes they begins translating the mRNA of the protein is destined for the secretory pathways. The first 5 to 30 amino acid polymerase encoded a single peptide the molecular masses that is recognized and bounded by the single recognition particle, that is SRP. The ribosomes that becomes attached to the endoplasmic reticulum, they synthesize all transmembrane proteins. So most secreted proteins that are stored in the Golgi apparatus, lysosomes, are and in the endosomes. The translation, the pause and the ribosomes complex binds to the rough endoplasmic reticulum's translocon. The important function of the endoplasmic reticulum is a transportation of proteins. So as the proteins are get formed in the endoplasmic reticulum, they are transported through the tubules towards the proteins of smooth endoplasmic reticulum that will lies near to the Golgi apparatus. So at this point, what will happen? The small transport vesicles, components of the small envelope of the smooth endoplasmic reticulum, will continually break away and diffuse to the deepest layer of the Golgi apparatus. So inside these vesicles are the synthesized proteins and other products from the endoplasmic reticulum is present. The transport vesicles, they are surrounded by the coating proteins, that is COP1 and COP2. So COP stands for coat protein complex. Okay, so this coat protein complex second, they target the vesicle to the Golgi apparatus. As I told you in the previous lectures, that whatever the protein will be synthesized, so they get protected by the vesicles and transported toward the Golgi apparatus. So this transparent proteins from the rough endoplasmic reticulum to the Golgi apparatus, and this process is termed as anterograde transport. Right? So such kind of transport is called the anterograde. The COP1, that is coated protein complex 1, transport the proteins from the cis end of the Golgi complex to the back of the rough endoplasmic reticulum. So this process is also termed as a retrograde transport, just opposite to the anterograde transport. The functions of the rough endoplasmic reticulum is this: they provide the surface for the ribosomes for the attachment, and they also provide the surface for the protein synthesis, as protein synthesis also takes place inside the endoplasmic reticulum, rough endoplasmic reticulum. Then the next function, that is the formation of glycoproteins, which are linking of sugars to for the glycoproteins, starts in the endoplasmic reticulum and is completed in the Golgi complex, right? Then the synthesis of precursors. RER, they produce the enzymes precursor for the formation of lysosomes by the Golgi complex. Smooth endoplasmic reticulum formation is also takes place by the rough endoplasmic reticulum. Then we will see the functions of smooth endoplasmic reticulum. So this three-dimensional structure of the endoplasmic reticulum shows smooth endoplasmic reticulum structure. So this smooth endoplasmic reticulum lacks the ribosomes and the function in the lipid metabolism, carbohydrate metabolism, and the detoxification. Right? So these are the major functions of endoplasmic reticulum in the mammalian liver and the gonad cells. It also synthesizes the phospholipids which are required for the cells. The cell which secrete these products such as the testes, ovaries, and the skin oil glands, they have great deals of smooth endoplasmic reticulum. Means they get formed from the smooth endoplasmic reticulum. The important function of this is the detoxification. Smooth endoplasmic reticulum brings the detoxification in the liver, which convert the harmful material, that is some drugs or poisons, into the less harmful form. Then formation of organelles, like the smooth endoplasmic reticulum produces the Golgi apparatus, lysosomes, and vacuoles also. So these cell organelles get formed from smooth endoplasmic reticulum. Then it also arises out the attachment of the receptors on the cell membrane proteins and the steroid metabolisms. So these are the functions of smooth endoplasmic reticulum. In the muscle cells, it regulates the calcium ion concentration. Muscles can interact, like contract as well as release. So this will be takes place because of the calcium ion concentration. So this calcium ion concentration also get controlled by the smooth endoplasmic reticulum in the muscle cells. Smooth endoplasmic reticulum also contains the enzymes like the glucose 6 phosphatase, which convert the glucose 6 phosphate to glucose. It's a step in the gluconeogenesis. Right? So today we'll stop here, and in this lecture we have seen the three different kinds of cell organelles: chloroplast, mitochondria, and the endoplasmic reticulum. In the next lecture video we will learn about the cell cycle. Thank you.